Morning, all of you. Good morning. Okay, now it is 40 students. Our present current topic is carbon and energy source. How to this particular the carbon is transmit to as a form of energy in all either in oxyphosphorylations or anoxyphosphorylation. This is a major difference. Oxyphosphorylation is takes place with water is it's carrying one. Here it is. Oxygen is releases anoxyphosphorylation. In this, instead of water, other gases, H2S, hydrogen, and other the sulfur reductants is used. This is the thing up to here we are identified. There and the next the classification wise. And also we have a lot of okay. Wait a minute. Okay, 1 minute. This is of our major classification. The energy, the source of energy, is in a carbon source of energy in living things, living organisms in biological way. It's in a chemotropes, it's in a chemical. This is a main component. And phototropes, the light source is the main component. Here it is the carbon source. Using CO2 and organic compounds. At the same time, in a phototropes also, the carbon, inorganic carbon content of CO2 and organic compounds containing of the carbon. In what way? You all of you, the lesson once, here it is, carefully. The next, it just converts of in a as a energy source wise how it is transmitted. Here you see the hydrogen, sulfur, and here it is the oxygen in photosynthesis and oxygenic phosphorylation, oxygenic phosphorylations, and all. Listen carefully.
We are going to discuss about the classification of organisms based on their carbon and energy source. So as we all know that all organisms require energy for their survival and growth. Okay. Now this energy might be coming from either a chemical source or a light source. Okay. If the energy is coming from a chemical source, then the organisms are known as chemotrophs. And if the energy is coming from a light source, then the organisms are known as phototrophs. Okay. Now, these chemotrophs and phototrophs can be further divided into two subtypes based on the carbon source. If the carbon source is CO2, then they are known as chemoautotrophs. If the carbon source is organic compounds, then they are known as chemoheterotrophs. Similarly, for phototrophs, if the carbon source is CO2, then they are known as photoautotrophs. And if the carbon source is organic compounds, they are known as photoheterotrophs. Now, the difference between this auto
Yesterday we had discussed up to here. The major component of the carbon source, the energy source of carbon is in organic compounds, natural carbon dioxide. And the other source is chemoautotrophs, chemoheterotrophs, photoautotrophs, and photoheterotrophs. But what is going on at here onwards? What way it distribute the energy in the form of up to ATPs in a cell. The energy causing substance of ATP is in what way it generates, either in aerobic or anaerobic. Is it phosphorylation or anoxyphosphorylation or an oxyphosphorylation? Wise, you find.
autotrophs, okay, both the autotrophs and both the heterotrophs is that the autotrophs use raw materials like CO2 and chemical or CO2 and light to produce organic compounds, and these organic compounds which are produced by this both these autotrophs are used by the heterotrophs, both chemoheterotrophs and photoheterotrophs. So autotrophs, okay, are producers in the environment, and heterotrophs are consumers which are dependent on the autotrophs for their carbon source. Okay. Now the chemoautotrophs include examples like hydrogen, sulfur, iron, nitrogen, and carbon monoxide oxidizing bacteria. Okay. Now let's move on to chemoheterotrophs. Now in chemoheterotrophs, the final electron acceptor can either be O2 or it cannot be O2. So based on that, it is classified further. Okay. If it is O2, electron transport chain which occurs in the membrane of the mitochondria, if the final electron acceptor is O2, then all the which includes all the animals, fungus, protist, bacteria, and also humans, then therefore we require O2. Okay. And if it is not O2, then it can be either organic compound. So yeah, in the case of fermentation, now fermentation like alcohol preparation or this curd preparation, it occurs in the absence of O2, and in that the electron acceptor are organic compounds. Now, other than organic compounds, the electron acceptor can also be inorganic compounds like nitrates, sulfates, etc. And the examples is Pseudomonas nitrificans. Okay. So now let's move on to the phototrophs and the photoautotrophs. So photoautotrophs can either emit oxygen or they don't emit oxygen. So if they emit oxygen, they are known as oxygenic, and if they don't release oxygen in the environment, they are known as anoxygenic. Okay. So if they use H2O to reduce CO2 and emit oxygen, then they are known as oxygenic. And if instead of H2O they use some other compounds like H2S or only H2, then they don't release oxygen, and then they are known as anoxygenic. So oxygenic photosynthesis occurs, as we all know, that in plants, okay, algae and cyanobacteria, and anoxygenic photosynthesis occurs in green and purple bacteria. Now, these examples of this photoheterotrophs include green non-sulfur bacteria and purple non-sulfur bacteria. So that's it for
Okay.
today, guys.
This is
Thank
of
you.
our classification. Chemoautotrophs, chemoheterotrophs. Chemoautotrophs is only hydrogen, sulfur, iron, nitrogen, and all. And here it is in chemoheterotrophs in oxygen, with oxygen or without oxygen. Already in nitrogen cycle, we are identified in absence of oxygen also the Pseudomonas bacteria, and fermentation is takes place. It is anaerobic, and at the same time, of our septic litter tank, whatever the bacteria is grown inside, the entire the fecal stands, it converts the waste substance the, as a waste sewage, sewage, sorry, waste sewage. In what way? That is nothing but of anaerobic. But here is a fungus, proteins, and protists, yeah. Already in archaea were identify the protists and the bacteria and fungi. These are all are requires in presence of oxygen. Here, in this case, the photoautotrope and photoheterotrophs point of view, instead of the water, it will reduce the carbon dioxide. It's already we discussed on yesterday. Oxygenic and oxygenic. Oxygenic photosynthesis, and oxygenic photosynthesis or phosphorylation. Oxygenic phosphorylation and oxygenic phosphorylation we are identifying. And next, organic um, compounds containing of the carbon. These are called as photoheterotrophs. It's in green non-sulfur the bacteria. The non-sulfur bacteria, the sulfur bacteria is majorly in coal mines where identify the sulfur bacteria. A lot of sulfur bacteria in anaerobic conditions it grows. There's a lot of very dirty odor. Dirty odor just comes out because of this bacterial load and sulfur. In sulfur also this particular bacteria can grow. Okay. Once again you see.
Energy source. So as we all know that all organisms require energy for their survival and growth. Okay. Now this energy might be coming from either a chemical source or a light source. Okay. If the energy is coming from a chemical source, then the organisms are known as chemotrophs. And if the energy is coming from a light source, then the organisms are known as phototrophs. Okay. Now these chemotrophs and phototrophs can be further divided into two subtypes based on the carbon source. If the carbon source is CO2, then they are known as chemoautotrophs. If the carbon source is organic compounds, then they are known as chemoheterotrophs. Similarly for phototrophs, if the carbon source is CO2, then they are known as photoautotrophs. And if the carbon source is organic compounds, they are known as photoheterotrophs. Now the difference between this autotrophs, okay, both the autotrophs and both the heterotrophs is that the autotrophs use raw materials like CO2 and chemical or CO2 and light to produce organic compounds, and these organic compounds which are produced by this both these autotrophs are used by the heterotrophs, both chemoheterotrophs and photoheterotrophs. So autotrophs, okay, are producers in the environment, and heterotrophs are consumers which are dependent on the autotrophs for their carbon source. Okay. Now these chemoautotrophs include examples like hydrogen, sulfur, iron, nitrogen, and carbon monoxide oxidizing bacteria. Okay. Now let's move on to chemoheterotrophs. Now in chemoheterotrophs, the final electron acceptor can either be O2 or it cannot be O2. So based on that, it is classified further. Okay. If it is O2 in electron transport chain which occurs in the membrane of the mitochondria, if the final electron acceptor is O2, then all the which includes all the animals, fungus, protists, bacteria, and also humans, and therefore we require O2. Okay. And if it is not O2, then it can be either organic compound. So yeah, in the case of fermentation, now fermentation like alcohol preparation or this curd preparation, it occurs in the absence of O2, and in that the electron acceptor are organic compounds. Now, other than organic compounds, the electron acceptor can also be inorganic compounds like nitrates, sulfates, etc. And the examples is Pseudomonas nitrificans. Okay. So now let's move on to the phototrophs and the photoautotrophs. So photoautotrophs can either emit oxygen or they don't emit oxygen. So if they emit oxygen, they are known as oxygenic, and if they don't release oxygen in the environment, they are known as anoxygenic. Okay, so if they use H2O to reduce CO2 and emit oxygen, then they are known as oxygenic. And if instead of H2O they use some other compounds like H2S or only H2, then they don't release oxygen, and then they are known as anoxygenic. So oxygenic photosynthesis occurs, as we all know, that in plants, okay, algae and cyanobacteria, and anoxygenic photosynthesis occurs in green and purple bacteria. Now, these examples of this photoheterotrophs include green non-sulfur bacteria and purple non-sulfur bacteria. So that's it for today, guys. Thank you for watching this.
Okay. Yeah. Now, these all are microorganisms. Microorganisms, majorly of the bacteria. What way this particular energy source is transferred. You find you listen carefully.
Organisms, and really all organisms, by what energy source that they use. An example being a phototroph. If you look at the word photo, as in photon, that's an indication that they're going to be using light, and it's usually sunlight that they use for energy. And we think of this as being plants and algae and certain bacteria that are able to use sunlight for energy. If they're not a phototroph, they're going to be a chemotroph, which means they're going to be using chemical compounds, and that could mean chewing up a dead organism and getting energy from the various components in them. It could be they're eating sugar or something like that. That's a chemical compound, and that's how they're getting their energy. We're going to find that pathogens fall into this category of chemotrophs. And of course, not just pathogens, we are chemotrophs.
Pathogens is comes under
dogs,
this
chemotroph,
category.
that sort of thing. Good bacteria happen to also be chemotrophs. Some of them are not chemotrophs. We do find bacteria that are able to
Someone
use
is healthy
sunlight as a source
bacteria,
of energy,
someone
and they're
harmful
phototrophs. So
bacteria.
we find bacteria in both groups. We can also classify organisms by what their carbon source is. An example being, we have something that's called an autotroph, and they can be referred to as self-feeders. The word auto oftentimes means self in Latin. And what that means is they're not depending on anything living for carbon. They're getting their carbon dioxide usually from they're getting their carbon from something like carbon dioxide, that sort of thing, or other inorganic molecules that contain carbon. Heterotrophs, hetero meaning different, meaning they are relying on something different for their carbon source. And so you could say they're depending on other life forms. Either they're eating the life form while it's still alive, or it's no longer alive. If you want to think about it, we eat plant material. That would be considered another life form, and we're getting our carbon from them. We're going to get, if you're eat meat, you get carbon from that. So they're using organic molecules. So that's the big difference. Inorganic molecules indicating that it's not alive for the autotrophs, and for the heterotrophs, it's utilizing organic molecules. And they can get that carbon from sugars, from proteins, from lipids, anything that contains carbon. We are going to find pathogens in this particular category. And so here we have a picture of our wonderful cat here. It says, just found out I'm a heterotroph. Go figures. And I just added that because I like cats. Now, we never do anything simply in science. We combine those terms. to describe organisms in manners of which describe what energy and carbon. So combine those two together. And if we're looking at a pathogen, the energy source for a pathogen, we would use the term chemo, that prefix. If we're talking about what source of carbon they're using, the pathogens would be hetero. And so we can combine those two and get what's called a chemoheterotroph. So I have a picture of a cat here. Yes, cats are, they're not pathogenic, but they are chemoheterotrophs. We are chemoheterotrophs, and many bacteria are chemoheterotrophs, especially pathogens, because pathogens are using us as a carbon source and as an energy source. So other terms that we can use to describe chemoheterotrophs can be parasitic. So if you think about it, that's an organism that would infect you and use you for both your carbon and your energy, but it's not a benefit to you at all. And so it's at our expense. And then a worm would be an example. And I threw in a couple of other cartoons
There's a
about
leech.
parasitic things here for you.
Leech sucks the blood.
And then another term is something that's called saprobic. And these are organisms that aren't parasitic, but they are breaking down material. And that's usually the kind of an organism that's going to be breaking down dead material and then taking the carbon and hydrogen and other nutrients from that and using it some for themselves and other it's releasing into the environment. So an example of saprobes, as I've indicated here, they're
Saprobes is
decomposers.
nothing but of
You can think
saprophytes.
of a lot of the fungi. If you were to go out in a forest and you see
Fungus.
that a tree has fallen down, you'll actually see mushrooms growing on them in some cases. And what you can't see necessarily are microorganisms that are also breaking them down as well. And here what they're showing you is kind of what saprobes do. These particular organisms, they can't necessarily get this bigger material that's out in the environment into themselves as a source because their cell wall acts as a really good barrier against it, but they want the material. So what they do is they produce these enzymes that can break down these bigger molecules into the parts that make it up. So if you can break up a protein into amino acids, and then those amino acids are much smaller and they're able to bring that in. And so that's what they do. And we especially see that with fungi. If you were to get spores from a fungi onto something like a blood agar plate, which has red blood cells in it, usually from a sheep, it will release some of these digestive enzymes, and then you get this zone around the fungal colony that's growing where it has destroyed all the red blood cells and taken up the nutrients, so it's clear around it. It's really kind of neat looking. But that's what saprobes are. So if you want to take a look at the whole big scheme of thing with microorganisms, we can break them up into either autotrophs or heterotrophs, and then that's breaking them up by the carbon source, either inorganic or organic. And then you can add to it whether they're using light as an energy source or if they're using chemical compounds as their energy source. So we do have photoautotrophs, chemoautotrophs, photoheterotrophs, and chemoheterotrophs.
Again, you
This
listen
class
carefully.
having a
These
focus
four types.
a fair amount on pathogens, what we're really going to be looking at a lot is going to be chemoheterotrophs. And so you can see here it's most bacteria, all protozoans, all fungi, all animals, that sort of thing.
Okay. This is for energy transportation. Okay. phototropes, the major is photosynthesis, the photons of energy to sunlight, whatever its usage. You see already carbon energy source wise, the phototropes, chemotropes, yeah, the lithotropes and organotropes. But here it is just only we are distinguished of inorganic and organic carbon. This classification is not at mentioned into there, but anyhow, there it is. The carbon dioxide is inorganic carbon, and other source of organic carbon is also there in that classification. But here, the usage of inorganic and organic carbon wise, photolithotropes, photoorganotropes, chemolithotropes, and chemoorganotropes. Either instead of the water, other oxygens or other gases, or else in presence of oxygen or in absence of oxygen. And at the same time, the chemolithotropes is totally, water is not at, there it is totally gases only. Hydrogen, sulfur, nitrogen, every gas is takes place. And chemoorganotropes were identified of aerobic and anaerobic. Here it is nitrogen fixation bacteria also takes place in our nitrogen cycle. Were identified, and phosphorylation bacteria. But the ultimate thing is it's in oxyphosphorylation or else an anoxyphosphorylation. The ultimate thing is it can produce of ATP through photophosphorylation. And the photophosphorylation is nothing but a photosynthesis. Through this only. Why? Because these are the autotrophs. They are the primary one, prime most. The next onwards, all are heterotrophs, dependers. The energy it begins from here onwards. Of course, the chemophotos and the chemophotosynthesis is a different story. That is all are comes under the microorganisms especially, and the pathogens also. This is the functionality goes on. You understand the major difference. But this in the phosphorylation, photosynthesis it begins the main energy source of conversion of carbon energy source. In what way? The major, the chemoorganotropes. Chemoorganotropes and organic compounds is its supply, both in carbon and energy point of view. But in all the cases of the metabolism means its activities of anabolism and catabolism. Anabolism and catabolism, the destruction and at the same time the cohesive structure mass generation. These two is that takes place. In what way? The first prime most one of this is the photophosphorylation, the energy transformation, photons of carbon energy of inorganic carbon.
Sprinkle
Usage
some
of carbon
rainwater,
dioxide.
you add some carbon dioxide from the atmosphere, and through
Inorganic carbon
photosynthesis,
dioxide.
plants will give off oxygen and will create simple sugars such as glucose. But you know, the
Which
actual
is
process
glucose
is a
or
lot more
sucrose,
complex.
C6H2O.
So let's get started. So the sun gives off a variety of types of energy, and this
Yeah,
energy
this is
travels
about
across
sunlight.
space in the form of a wave. and the titles of the energy you can see at the top of the picture. Now we're going to focus our attention on visible light because it's the wavelength. It might seem pretty straightforward. You shine some sunlight, you sprinkle some rainwater, you add some carbon dioxide from the atmosphere, and through photosynthesis, plants will give off oxygen and will create simple sugars such as glucose. But the actual process is a lot more complex. So let's get started. So the sun gives off a variety of types of energy. This energy travels across space in the form of a wave, and the titles of the energy you can see at the top of the picture. Now we're going to focus our attention on visible light because it's the wavelength of visible light that drives and powers photosynthesis.
Visible light is used only for photosynthesis.
Let's focus on how light is absorbed. And when we do, we're going to see that the sun gives off visible light, also called white light. White light is actually a mixture of Roy G. Biv. And what this is are the colors of the rainbow: red, orange, yellow, green, blue, indigo, violet. All these colors are a mixture known as white light. Well, how do we know this? Because when you shine light into a prism, when you shine white light into a prism, a rainbow comes out the other end. Just like you can see in this animation. White light is entering from the left, and because of the curvature of the prism, Roy G. Biv, the colors of the rainbow are coming out the other end. Notice how the wavelengths are different. Red has a longer wavelength, violet has a more narrow. And so this is how we know that white light is made from a mixture of these colors. Here's an actual picture of white light entering a prism, and you can see on the left-hand side a rainbow is coming out the back end. Moisture in the air can act like a prism and create a rainbow. You ever notice after it rains, you can sometimes see a rainbow in the sky, or in this case, near a waterfall, because of all the moisture in the sky. The moisture separates the light, the sunlight, into the rainbow colors that you see. And so colors are either reflected or absorbed. When sunlight shines on an object, what we see is reflected light. The reason this leaf appears green is because it's reflecting the green wavelength of light. When light shines on these bananas, the reason it appears yellow is because the yellow wavelength reflects, while the other colors are absorbed.
So this is a
When we
fundamental.
shine
Whatever the color
light these
is
balloons,
visible,
the blue
reflects.
wavelength reflects
The rest
into
of
our
colors
eyes,
are
and the other
absorbed.
colors are absorbed. When we look at white objects like snow or this Arctic hare, the reason object white is because white reflects all the colors, and all the wavelengths of light enter our eyes, and our brain interprets this as the color white.
But
Well, the opposite
here,
would be black.
all
When
are
light
absorbed.
shines on a black
No
object,
reflections.
nothing reflects. It all is absorbed, and that's why this automobile appears black, because there's no light reflecting into our eyes. So when we relate this to photosynthesis, plants have mostly green leaves, which means that they're going to reflect the green wavelength, but the other colors are absorbing into the leaves and are going to be powering photosynthesis. In this
The
graph,
visible light
you can see,
wavelength,
for instance, that
352,
this graph shows the
750
wavelengths of light that most power photosynthesis. Notice how there's a peak around 400 nanometers. Well, that's the wavelength of blue. There's another peak around 700 nanometers. Well, that's the wavelength of red. So blue light and red light are most stimulating for plants in the process of photosynthesis. Ironically, the color green around 550 nanometers, the color green that we most associate with plants, actually has the least amount of impact in their ability to do photosynthesis. That's because most green light is reflecting. Very little green light is actually absorbing into the leaf. So when we look at a general definition of photosynthesis, it's the process that converts solar energy into glucose. And this formula here outlines photosynthesis. And in the reactants, we have carbon dioxide and water. And in the presence of sunlight, we'll produce sugars such as glucose and oxygen. Well, who performs photosynthesis? Well, these are the autotrophs. Now, we typically think of plants as doing photosynthesis. But they're not the only ones. Algae, phytoplankton, the basis of ocean food webs, kelp. Kelp can grow into these enormous underwater forests, and even some bacteria known as
Yeah,
cyanobacteria
underwater
are
plants. The
photosynthetic.
rest
So
of
there's
one,
a
you
lot
know,
more autotrophs
all
than
the
just plants. So photosynthesis is also what we call an endergonic chemical reaction. And what that means is that photosynthesis requires the input of energy. From the picture, I'm implying that sunlight is the energy that is input to power photosynthesis.
yeah, you remember end organic
And when we
chemical
talk
reaction.
about the end result, what's created? Well, oxygen, which is a waste gas, and simple sugars like glucose. But when we zoom out into the leaf, we find some peculiar pores on their underside. These pores will open and close, and they're called stomata, and it's how they exchange gases. It's how they take in carbon dioxide from the atmosphere.
Here it is
And when they
carbon
produce
dioxide
oxygen as a
exchange.
waste through these stomata openings, they give off the oxygen created. So it's through these pores that they exchange gas with the atmosphere. Okay, so now let's introduce the structure in cells that allows them to do
This is about
photosynthesis. And this is the
chloroplast.
chloroplast. What we're going to do is we're actually going to divide photosynthesis into two stages. Stage one being the stage that depends upon sunlight, the light-dependent reactions. And these take place in the thylakoid membranes of a chloroplast. You know, I want to actually look at a more simplified diagram here. And when we do, we can actually see the thylakoid frisbees stacked on top of one another. Well, in the membrane of these thylakoids is where the light-dependent reactions occur. And if you ever come across the term granum, that's a term that means
Granum
a stack
and stroma.
of thylakoids.
The thylakoids.
Okay, so you see that flashing black box? I want to zoom into that area of the chloroplast. And when embedded within the membrane of the thylakoid, I've labeled them PS1 and PS2, and these are the photosystems, and we're going to talk about them in more detail, but for now, in the photosystems is where we find chlorophyll and various protein molecules that are responsible for capturing and transferring energy during the light-dependent reactions. And it's during the light-dependent reactions where oxygen, ATP, and something called NADPH is created. We'll talk about these molecules in more detail coming up very shortly. And then we'll shift into stage two, the light-independent reactions. And this happens in the stroma, the fluid-filled interior of the chloroplast, just outside of the thylakoid. And it's in stage two, the light-independent reactions, where glucose is actually created. So let's talk about these two stages for the rest of this video. So now let's get into some of the details of the light-dependent reactions. And what we're going to do is we're going to break this down into a few steps. So step number one, chlorophyll in photosystem number 2 will absorb sunlight. Now, even though it's called photosystem number 2, it's actually the first in the chain of events. They were named photosystem 1 and photosystem 2 by order in which they were discovered, not the order in which they perform their functions. So you kind of have to get over that backwards name right there. So in my animation, sunlight is striking the chlorophyll molecules in photosystem number 2, and this causes their electrons to become excited. And what happens is the electrons actually flow into the thylakoid membrane. And so as these electrons from chlorophyll in photosystem number 2 flow through the thylakoid membrane, this makes the membrane negatively charged. And as the electrons are flowing through, there's also proteins embedded within the thylakoid membrane that are aiding their travel. I just didn't show them in my animation. But this starts what we call an electron transport chain. It's a chain of events set in motion by these electrons. So when we move on into the second step, I've added some hydrogen ions into the stroma because there's dissolved molecules and ions in the stroma, and these hydrogens are going to play an important role. And so what happens is, as the electrons move through the thylakoid membrane, they actually will operate in power and turn on these protein pumps that will pull the hydrogens into the thylakoids. And as more and more and more hydrogens accumulate, this is going to make a really large concentration of hydrogens, which we'll see why that's important in a little bit. I also want to mention that water molecules are broken. So here's a water molecule, and with the help of enzymes, the hydrogens are broken off, and the electron from the hydrogen went into photosystem number 2. Here we go again. Enzymes will break off the hydrogen, and notice how the electrons were pulled into photosystem number 2. You know, photosystem number 2 has been losing electrons, and so it's water's, the hydrogen of water, it's their electrons that will replenish photosystem number 2 so it doesn't run out of electrons, and so this process can keep
Whatever
going.
the
This is
water
why
is
you have to
consumed,
constantly water a plant.
yes, water is its
Also, oxygen. This is where the oxygen
Great,
comes
that's
from. See
photosystem
that
too.
O for oxygen? It will bond with another O from another
But this
water that was
particular
broken to
water
make
is xylem and
O2.
phloem
The
is
oxygen that we
transferred.
breathe used to be attached to water, which I think is kind of interesting. So, as we move on into the next step, again, sunlight is striking photosystem number 2, and its electrons are moving through the thylakoid membrane. But you know, sunlight is also striking photosystem number 1. And photosystem number 1 is filled with chlorophyll, and it has electrons that also become excited and move through the thylakoid membrane. So what we really have is kind of a longer electron transport chain. So now that we have our full electron transport chain, let's move on to the next step. And in the next step, a molecule called NADPH will be created. So in the stroma, NADP and a hydrogen are drawn together. And the electrons of the electron transport chain help these two bond together to make a really important molecule called NADPH. NADPH can be kind of be viewed as a carrier of hydrogen, like a taxi whose job it is to carry hydrogen from one place to another. We'll come back to it in a little bit, but it's really
Nitrogen
important that
amino-diphosphate.
NADPH was just created.
NADPH,
So at
phosphohydrate,
this stage, the
NADPH.
hydrogen is really beginning to accumulate inside the thylakoid from the breaking of water, from the electron transport chain, pulling in hydrogens from the outside. So by now, there's a really large concentration of hydrogens inside the thylakoid. So when we come to our next step, because there's such a large amount of them, the hydrogens are going to start to diffuse through that enzyme labeled ATP synthase. And so what hydrogen does from a high to low concentration, it'll diffuse through ATP synthase and will help to bond a molecule of ADP, adenosine diphosphate, with another phosphate. And when that happens, they bond to make a molecule of ATP. And so this ATP, along with the NADPH,
It's a triphosphate,
are really the
three
most
Ps.
important things that are created during the light-dependent reactions. We're going to see what's so important about
ADP is
them
a
in just a little
diphosphate.
bit. And so to summarize the light-dependent reactions, oxygen, NADPH, and ATP have been created. You know, sunlight was taken in, water was taken in, and oxygen was released as a waste. And in the light-dependent reactions, NADPH and ATP have been created. I do want to note at this stage, no glucose has been created. So let's move on to the light-independent reactions. Okay, so the light-independent reactions. You know, this is also known as the Kelvin cycle, named after the scientist who helped to identify these steps. And so we're going to start breaking it down. First of all,
There's a Kelvin
there's a
cycle.
5-carbon molecule by the name of RUBP,
Light
ribulose
independent reaction is Kelvin
biphosphate.
cycle.
Now, it has more than just 5 carbons. You can see 12 hydrogens, 11 oxygens, and 2 phosphoruses. But for simplicity, I've only drawn the 5 carbons of RUBP. And what happens is RUBP bonds with a molecule of carbon dioxide. And when they bond, they
Ribose
make a very
biphosphate.
short-lived 6-carbon molecule. The 5 carbons from RUBP plus the 1 from carbon dioxide make this very short-lived 6-carbon molecule. So the reason the 6-carbon molecule is short-lived is we finally get to see the importance of ATP and NADPH, which were created during the light-dependent cycle. The 6-carbon molecule is going to be broken down by the energy contained within ATP and NADPH. And enzymes will help to break down ATP and NADPH to release their energy. And when the energy is released, that 6-carbon molecule is broken down into 2 molecules called phosphoglycerate. Each PGA or phosphoglycerate is 3 carbons in size. And by the way, the NADP and the ADP that are left over will be reused and recycled in the next light-dependent reaction. And so this happens multiple times. Here's another short-lived 6-carbon molecule broken down by NADPH and ATP to make
Again,
2 more molecules of phosphoglycerate. And here's another 6-carbon molecule broken down by another NADPH and another ATP to make 2 more molecules of the phosphoglycerates.
Number of
So what happens
phosphoglycerates
now with
generates.
these phosphoglycerates? Well, some of those 3-carbon phosphoglycerates are going to bond with one another, and with the help of enzymes, they're going to bond to make simple sugars, such as glucose. And that's the whole
This is
point
ultimate
of
product.
photosynthesis, is to make
C6H12O is
glucose. Now,
fructose.
not all the phosphoglycerates do this. Some of the phosphoglycerates are going to help to keep the cycle going. And so what happens is they're going to be broken down. They're going to be broken down with the help of ATP to keep the cycle going. And when ATP breaks some of these other phosphoglycerates down, they're broken down. And then enzymes help to build that same 5-carbon molecule we saw earlier, ribulose biphosphate. And now we're repeating the Calvin cycle, the light-independent reactions. The process simply restarts because carbon dioxide will bond with this 5-carbon RUBP. When they bond, they make that short-lived 6-carbon molecule that we mentioned earlier. And the process repeats itself. So to summarize the light-independent reactions, carbon dioxide, ATP, and NADPH are going to create glucose. So when carbon dioxide is added to the ATP and NADPH from the light-dependent reactions, with the help of enzymes, you have simple sugars such as glucose created. Okay, so if you're in my biology class, we'll talk about this essay question in class.
Yeah.
And there you have it. As we wind down this video, pause it, try to answer these